Thanks, Silka. So this audience would know that cells use a variety of different calcium channels and pumps to achieve specific cellular outcomes. But cancer cells can also use those same calcium channels and pumps to achieve what they need to do to contribute to tumorigenesis. So for example, in this diagram here, we look just at breast cancer. There's a variety of different calcium channels and pumps that can be linked to breast cancer proliferation, cell death and resistance, metastases, and even the new emerging field of the tumor microenvironment. So we know some channels can regulate those processes. So if we step back and think about, well, how could we identify what calcium channels or pumps we should work on when we're thinking about a therapeutic target? And really, if we oversimplify the process, there's two mechanisms that we can use. One is to look at the overexpression or try and find calcium channels and pumps that are significantly overexpressed in particular cancers or particular cancer subtypes. And the other arm that we could use is to identify what calcium channels and pumps contribute to processes important in tumorigenesis. And I want to start on the second part first in terms of how can we use that approach to identify potential therapeutics. So one way is what we've looked at is cellular plasticity. So cancer cells need to go sometimes to metastasize from a more epithelial type approach, which is that they're very proliferative, to a mesenchymal approach where their phenotype is much more migratory, they're able to spread, they're also able to often resist therapeutics. So one of our PhD students, when she started, Felicity Davis, who now has her own research group at Huss University, she focused on this phenomenon of epithelial to mesenchymal transition, which was mentioned earlier in the session previously. So what Felicity demonstrated is the calcium signal is a key event in the induction of EMT. So for cells to go from an epithelial morphology to a mesenchymal one, calcium is a key trigger in that event. And she also showed that was a massive remodeling. Epithelial cells have very different calcium features than migratory mesenchymal ones, even though they come from the same cell. When they're induced in the different phenotypes, they have different calcium signaling pathways. And so Felicity demonstrated with her model in 468 breast cancer cells that EMT was calcium dependent, that purinergic receptor signaling was majorly remodeled, and that TRPM7 could actually control the induction of EMT. And a variety of groups around the world have now shown that EMT is calcium dependent, and different cancer cells seem to use different calcium channels to induce their EMT status. What I want to focus on some work that just got published this year, which is a different phenomenon when we think about cell plasticity, and that is shape. So as epithelial cells are very rounded, whereas mesenchymal cells are sort of more stretched out, spindular. So Chun Leng, during her PhD with us, she's now at Johns Hopkins, just wanted to ask the question, what happens when we change the shape of a cancer cell? What happens to calcium signaling? And so she was very focused on cellular geometry. So how she did that is she used micropattern plates. So that she took MCF7 breast cancer cells, which generally are more epithelial-like, and she forced them into extremes of cell morphology. So she forced them to be rounded, or she forced them to be triangular, which is more representative of the mesenchymal phenotype. And what she observed is that there was an upregulation of mesenchymal markers when you made the cell more mesenchymal in shape. Now that was known, so that was our positive control, that when you change the shape of these cancer cells to a more mesenchymal shape, you had an upregulation of mesenchymal markers like fermentin and serpin-1 and snail. To our surprise, really, when we looked at calcium channels actually changed more than the mesenchymal markers. So we got a massive upregulation of some of the TRP channels, and particularly of interest was piezo-1 because that's a force-activated channel. So what we're thinking of, that might be particularly significant when we think about metastases, because a cell needs to escape the primary tumor, squeeze between cells, exposed to a lot of different forces, and here we're seeing an upregulation of piezo-1 just as a consequence of cell shape. So what she did was to say, well, what happens now when we activate piezo-1? So of course, she used the piezo-1 activated GOTA1, And she did a high content imaging experiment with GCAMP6. So here we can see that we have our cancer cells with different morphologies, the rounded ones and the triangular ones. And this is an orthogonal view. This is relative calcium on a pseudocolor scale over time. And what she found is that when you stimulated these cells, the rounded cells were much less responsive to iota compared to those triangular cells. And when she looked across multiple experiments, performed the different passages, when she looked at area under the curve, that sustained calcium influx from iota-1 was much more pronounced when the cells had an L-shaped morphology. So it seems like when you force cells to be more mesenchymal, they express mesenchymal proteins, but they also have an upregulation of the force channel piezo-1, and the cells seem to become more sensitive to that channel. So then she took cells and said, okay, what happens when we take cancer cells and we activate piezo-1? So because with Felicity's work, we knew calcium was important in EMT, we took 468 breast cancer cells and added piezo-1 activated iota, and we saw an upregulation in vimentin expression, which is a mesenchymal marker, and a downregulation of ECAD, which is an epithelial marker. Now, those are not as pronounced, even though it's significant, as growth factors that we've studied. But what did change at the same level was this SERPIN1, a protein really important in breast cancer metastases. And we observed that there was a really massive increase in SERPIN1 levels when we activated these cells with iota-1. So what that tells us is that certainly morphology can drive behavior, and maybe iota-1 might be an interesting target for some aspects of breast cancer. So another way I want to focus on now is the overexpression approach, trying to find calcium channels that are overexpressed in different cancer types. And so one way that we could exploit an overexpression of different cancer types is by using an activator. So calcium is a trigger for cell death. So one hypothesis is maybe we can find a calcium channel that's so overexpressed that when we activate it, we actually can kill the cancer cell. So we had a lot of trouble finding such a channel. So we actually just developed an artificial system. This was Tina Wu doing her PhD. We took MCF7 cells and put TRPV1 in the control of a tetra-regulated system. When we overexpress TRPV1, you can see that the cells have become super sensitive to death induced by the TRPV1 activator capsaicin. But that's an artificial model. It took us a long time to find a channel that might be overexpressed to such an extent that we could use that in a real system. So we collaborated with Michael Mitski, who's a bioinformaticist from WeHi in Australia, and he looked at the calcium channel TRPV4, and he found that TRPV4 was really overexpressed in these basal molecular subtypes of breast cancers. And these are breast cancers that have poor clinical outcomes and in which there's the greatest need for new therapeutics. And so what he found, as we haven't seen this very often, is that some breast cancers had extremely high levels of TRPV4. And so we looked at breast cancer cell lines and found that indeed there were some breast cancer cell lines that had massively elevated levels of TRPV4 expression. So we took one of these cancer cell lines which overexpressed TRPV4 and asked the question, what would happen if we activated it? And this actually was work done with Silke when she was an undergraduate with us in our team. And what Silke did was live cell imaging and activated TRPV4 with this compound GSK. And what she observed was two different cell types. Two types of cell death. One was oncosis, where the iron gradient is so great the cell literally pops and explodes. And the other one is apoptosis, which is cell blebbing, which you're probably more familiar with. And Silke went to a lot of effort to quantitate that. And what she found was that the more you acted the TRPV4, the more likely we have to have this oncosis type of death, this massive increase in change in iron gradient that causes the cell to pop. Now, during his PhD, John Bassett, he had a PhD focused on methods. So he wanted to combine high content imaging, genetically encoded calcium sensors, and measuring calcium over extreme time periods over 48 hours. So John naturally was snapped up by Merck, and he's now working in Boston in that drug company. But before he left to work for Merck, he basically repeated Silke experiments with GCAMP6. And because he was doing a high throughput scenario, he could see cells do weird things next to each other. So here's what we see what Silke observed, which was when we activated TRPV4, we get, in this case, two different kinds of cell death. The popping, which is oncosis, which is a sudden increase in PI, and apoptosis, which is blebbing. And he's characterized that they're associated with different calcium phenotypes. Oncosis is a much more sustained calcium increase over that period. So you can theoretically target activators, but it's very hard to convince drug companies that that's a therapeutic approach. I think we can talk about it's viewed as a bit risky. So people generally focus on inhibiting something in cancer.